Recent research has highlighted the potential of Menadione, a synthetic precursor to vitamin K, in combating prostate cancer. A study led by Professor Lloyd Trotman at Cold Spring Harbor Laboratory discovered that Menadione induces cell death in prostate cancer cells by depleting a lipid called phosphatidylinosyl-free phosphate Pi3P. This depletion disrupts the cell's ability to recycle essential materials, ultimately causing them to rupture. In mouse models, this intervention significantly slowed cancer progression. CSHL.edu. These findings suggest that Menadione could be a promising therapeutic agent for prostate cancer. The research team is planning clinical trials to evaluate its efficacy in human patients. Additionally, the study indicates that Menadione may have potential applications in treating myotubular myopathy, or rare genetic disorder affecting muscle development in infants. CSHL.edu. It's important to note that while Menadione is a synthetic compound, natural forms of vitamin K, such as phylloquinone K1 and menaquinones K2, are found in various foods and have been associated with health benefits, including potential anti-cancer properties. However, the specific effects and mechanisms can vary between different forms of vitamin K. PMC.NCBI.NLM.NIH.gov. As with any emerging treatment, further research and clinical trials are necessary to fully understand the safety and effectiveness of Menadione in cancer therapy. Vitamin K precursor Menadione and cancer researchers and studies have revealed that Menadione vitamin K3, a synthetic precursor to vitamin K, has anti-cancer properties, particularly against prostate cancer. Research led by Professor Lloyd Trotman at Cold Spring Harbor Laboratory has shown that Menadione disrupts cancer cell survival mechanisms, leading to their destruction. How does Menadione kill cancer cells? Menadione targets and depletes a key lipid called phosphatidylinosyl-free phosphate Pi3P. This lipid is essential for a process called autophagy, which cancer cells use to recycle nutrients and survive under stress. Without Pi3P, cancer cells lose their ability to recycle resources, leading to cellular starvation and rupture. In animal models, Menadione slowed prostate cancer progression significantly. The study suggests Menadione could be developed into a new cancer therapy. Potential for human cancer treatment. This discovery is important because prostate cancer cells are known for their resistance to treatments like chemotherapy and hormone therapy. If further research confirms its effectiveness in humans, Menadione could offer a new targeted approach to killing prostate cancer cells. Clinical trials are being planned to test its effectiveness in human patients. Researchers are also exploring whether Menadione can be used for other cancers or diseases, including myotubular myopathy, or rare genetic disorder affecting infants. Vitamin K in cancer prevention and treatment. Menadione is a synthetic form of vitamin K, but natural forms vitamin K1 phylloquinone and vitamin K2 menaquinones are also being studied for their potential anti-cancer effects. Vitamin K1 found in green leafy vegetables may help regulate cancer cell growth. Vitamin K2 found in fermented foods and animal products has been linked to a lower risk of prostate and liver cancer. While Menadione has shown promising results, it is not yet approved for cancer treatment, and more research is needed before it becomes a standard therapy. Recent research has highlighted the potential role of vitamin K2 menaquinone in cancer prevention and treatment. Vitamin K2, a fat-soluble vitamin found in foods like meat, cheese, and fermented products, has been studied for its effects on various cancer cell lines. Mechanisms of action: inhibition of cancer cell growth. Vitamin K2 has been shown to suppress the proliferation of cancer cells by inducing cell cycle arrest, promoting differentiation, and triggering apoptosis programmed cell death. These effects have been observed in cancers such as hepatocellular carcinoma (HCC), leukemia, and pancreatic cancer. PMC.NCBI.NLM.NIH.gov. Induction studies indicate that vitamin K2 can induce autophagy, a process where cells degrade and recycle their components. This mechanism has been particularly noted in liver cancer cells. PMC.NCBI.NLM.NIH.gov. Inhibition of invasion and metastasis. Vitamin K2 has been found to suppress the invasive behavior of cancer cells, potentially reducing the risk of metastasis. This is achieved through the modulation of signaling pathways involved in cell migration. pmc.ncbi.nlm.nih.gov. Epidemiological evidence. Observational studies have suggested a link between higher dietary intake of vitamin K2 and a reduced risk of certain cancers. For instance, increased consumption of vitamin K2 has been associated with a lower incidence of prostate and lung cancers. Reuters. Clinical implications. While these findings are promising, it's important to note that much of the research is preliminary. Further studies, including large-scale clinical trials, are necessary to confirm the efficacy and safety of vitamin K2 supplementation in cancer prevention and therapy. Additionally, the optimal dosage and potential interactions with conventional treatments need to be established. In summary, vitamin K2 exhibits potential as a complementary approach in cancer prevention and treatment through various mechanisms, including inhibition of cell proliferation, induction of autophagy, and suppression of metastasis. However, more rigorous scientific investigations are required to fully understand its role and application in oncology.